you should shut the door.
It is. You know.
Okay. So like I said, we just left off with functional groups, and now we're going to talk about isomers. Isomers have the same molecular formula, but they have completely different structures. So I do have some examples of this. There are different types of isomers. You can be a structural isomer, which means that you got all the same atoms, but they are in different places. Okay, so they are rearranged in a completely different order. Or you can be a stereoisomer, which means you have the same carbon skeleton, but then those special groups that attach on the ends or on the sides are rearranged. And so there is a special stereoisomer called enantiomers, which means that they are mirror images. And so when I talk about left-handed and right-handed molecules, that's basically what I'm talking about. They are mirror images of each other. They look the same. I mean, they look in, you could take a molecule and show its reflection from a mirror. I mean, it would see that enantiomer to it. So a little flow chart of where I want to go. I'm going to talk about structural isomers. And so here we have a 3-carbon chain. I know it's probably blurry. But then they're emphasizing a fluorine, how the fluorine's attached on the end of this bottom one and attached to the middle carbon on this top one. So they have the same number of carbons, same number of hydrogens, same number of fluorines, but it's arranged in a different way. So that's a structural isomer. Over here we have our stereoisomers, and the only special stereoisomer I'm going to talk about is the antisomers. Oh my gosh, I hate that word so much. But this is showing reflection. So here's this molecule. This dashed line represents a mirror plane, and if it were to look, you would see the reflection of it. The chlorine, the fluorine, I can't even read that, and then the hydrogen's on the top. There are even more types of stereoisomers, but we will not cover them, so we're just going to talk about the category right here.
Wait, what's that called?
Enantiomer. Enantiomer. Oh my gosh, I don't know. I'm sorry. That's a, thank you. So, yeah, in college bio, I can probably only pronounce maybe like 70% of the words. I can spell it, but can't say it. Okay, so structural isomers, some more examples. Here I have pentane, the prefix 5. So C5H12 has 3 different types of structural isomers. So remember, structural isomers, same formula, but different structure. So here, 1, 2, 3, 4, 5. Got the same number of hydrogens, 12, okay? Or you can be a straight chain, or branched. So I have 4 down here, 1 carbon branched off. Okay, so structural isomers. So this diagram does the more 3D representation of the straight chain, the branched, and the multi-branched. So it is pentane, but because the carbon are placed in different places, obviously, now that we have a different name. So I took an organic chemistry in high school. It was called organic chem, okay? But really, it should have been called nomenclature 101, because all we did was learn how to name carbon molecules. So pentane, we're like, okay, pent, 5, and if it ends in A-N-E, single bond carbons, all the way through. So, boom. And then iso, prefix, means that there's 1 branch. So it's like isoformation in football. We have the fullback and the running back. So that's how you're supposed to remember iso. And then neopentane is like cross. So, yeah.
You said in the drawings yesterday, the one that just the lines drawings, you know, the
Yep.
stick, whatever,
Mhm.
that the line means carbon, right?
Yep.
Yeah.
Yeah, so like I said, so if you just saw this, that would be pentane.
Oh. So all that does is measure the amount of carbons.
Yeah, each point
Oh, it's hydrogen.
is a carbon.
Oh, carbon.
And if you don't see any branches, you assume it's all hydrogen. But then if you see this, okay, now I know there's a oxyl group, hydroxyl group on one end.
Wait, so what's the difference between the neopentane then? How did you remember that again?
It was just a cross.
Oh.
You don't need to. You just need to know what structural isomers are. That's it. These are just examples of the different structural formations. Okay, now I want to talk about naproxen and propanoic acid. So these two compounds have the same molecular formula. I mean, they have the same number of carbons, same number of oxygens and hydrogens, but they do differ in how their atoms are arranged in 3D space. And as a result, they are two completely different compounds. Naproxen is actually a pain reliever, and propanoic acid is a very smelly odor that your body can also give off. And then sometimes they use propanoic acid for like cheese flavors too. So kind of weird. I'm going to talk about naproxen later on in the next couple slides here. So the subcategory of stereoisomers and enantiomers, mirror images of each other. If you see the word chiral, that's talking about mirror images, the left-handed, right-handedness of a molecule. And it has everything to do with polarized light. It has a single plane, and so chiral molecules can either rotate left or right from this plane. There are two forms. You could be left-handed or right-handed. So D is right-handed and L is left-handed. So dextra and levomotory. Cool thing about left-handed, right-handed molecules is that in nature, one form is made, but you can manufacture it to be, the other handed, if you will. So like, great example here. I wish Logan was here so I could, because he asked about this. D sugars and L amino acids. So like D sugars come in two forms, left-handedness or right-handedness. And your body can only digest I think it's the right-handedness. It doesn't digest the left-handedness.
And the right hand is the dextra.
Yes. Yeah, sorry. Okay, I just want to go back real quick and look at something. Okay, so in this diagram, this is showing the sugars, the D glucose and the L, so the left and right-handedness of these two molecules. Yeah, so it's the D that's digested in our body and the L that's not. But they both still taste sweet. It's just that you can't digest the one on the right. Okay, so. Right. Oh, well, the reason why we can't digest it is because the enzymes in our bodies don't recognize it. So like when we talk about enzymes, they're very shape specific, and they can only, read or attach to molecules in a certain way. So then if you mix this up, the enzyme doesn't recognize it as a right-handed glucose. So therefore your body doesn't digest it. So a lot of artificial sugars can be just the opposite form of the original sugar. And so as a result, your body doesn't digest it. So you still taste the sweetness, but then you get the 0-calorie effect because your body doesn't digest it.
So is that like aspartame?
Sort of. Not.
I just know
It's
that's
a-
diets.
Right. It's a little. It's a variation of the original sugar molecule.
So what's the high fructose corn syrup?
It's just.
Is that right or left-handed?
I don't know. Actually, I really don't know.
Probably really too broad.
But I'll be happy to look it up. So, high fructose corn syrup handedness. Okay, so one more diagram here of stereoisomers showing the mirror image. So here I'm looking at the amino acid alanine. Got a carbon, COOH group at the top, a hydrogen, a methyl group, and some type of amine nitrogen group. Look into it in the mirror, it's just opposite of each other. But if you can't superimpose, you can't put the two on top. You can't take your left hand and your right hand and say, oh yeah, they're like perfect matches to each other. No.
So in the structural isomers that we already looked at, obviously, those ones are the ones that have are either straight lined or branched or what was the third option? Straight line.
Neo, crossed. Yep. So recap of what I just went through. Stereoisomers. Okay, we didn't talk about geometric isomers, but we did talk about enantiomers. Whew.
Wait, are stereoisomers and structural isomers different?
Say that again.
Are stereoisomers and structural isomers different?
Stereos ones are all are the weird ones.
Yeah.
left and right. Structures,
Oh, so those are like
different like shapes.
Okay, so real life example here with chiral left-handed, right-handed molecules. This is like probably my favorite example of left-handed, right-handed molecules and how they can have different properties. So limonene, two forms with almost the same structure as you can see at the top here. Okay, but when you look at them, they are mirror images. I have this ring-like structure with a methyl group on top, carbon with two hydrogens and a methyl group. And over here, that's where the methyl group is.
It's just that little line.
Yeah, double bond to the carbon with the hydrogen, and then methyl group on top. So, anyways, the right-handed form is found in oranges and lemons. Okay, and then the limonene is found kind of like in the skins of oranges and lemons as well, but it has a different property to it. So the right-handed is the orangey, smelly scent that you can smell, and the left-handed is lemony.
So the right hand is that the one on the right?
No, this is right, this is left.
So it's the positive one, at least the right one.
Well, yeah, because that's another way to do D and L. They have so many different forms, they can't just make up their minds. So. I know. Okay. Another example is, and there's no picture of it, but caraway. So you find this in Trident gum. So right-handed has the odor of spearmint, but the left-handed has the odor of caraway. And I always do like a fun fact trivia. I did one last year, and it was about Trident. What are the 3 flavors in the original Trident gum? Because it's the official gum of the White House, in case you didn't know. But does anyone know? It's like spearmint. What else is in there?
winter
No,
peppermint.
it's peppermint. And then caraway. Hey, he's here. And you just missed my whole spiel about left-handed, right-handed molecules.
Literally just that 2 seconds ago.
Okay, switching gears now. Okay, now we're going to talk about carbs and how we can make very big polymers. So, biological macromolecules include those carbs, nucleic acids, proteins, and lipids. And these macromolecules are polymers. They're large molecules. You make them by linking a bunch of monomers together. So here's my monomer, white, black. Well, if I get a bunch of these monomers linked together, I can form a large polymer. Or maybe each one of these is a monomer linked together by covalent bonds, and the end result is that polymer. So the monomer is the small subunit, polymer is the large molecule as a result of it. Polymers are made up of monomers. And so these are the 4 polymers we're going to talk about this week, and today we're going to emphasize carbohydrates. And the monomers of carbs are simple sugars or a glucose molecule. So how do we form polymers? Well, we got to undergo a chemical reaction. And so there's 2 different types of reactions I want to talk about. Dehydration reaction builds chains and forms bonds. So here I have my starting polymer. So monomer 1, 2, 3 linked together. I want to add this monomer to that chain. There's usually a hydrogen atom by itself, and this unlinked monomer usually has a hydroxyl group, and what happens is we're going to link it together by forming water. That's why it's called the dehydration. Okay. So then water forms as a result. I now have a covalent bond between monomers 3 and 4. So dehydration is also known as a condensation reaction because you're forming water, and when things condense, you see water.
So if you would have, this is not going to be an exact accurate example, but if you get what I'm saying. So if you like really hydrated the thing, would it like break everything apart?
Really hydrated the thing.
Because you said it's a dehydration reaction, so like a hydration reaction, would that be like breaking everything up?
Yes, it's the opposite, which is the second form of the reaction. Yes. Yeah. Very good. So steps for a dehydration condensation reaction, you're going to remove the OH from one monomer, the H from the other monomer, and you're basically just removing a molecule of water to form that covalent bond. Now the opposite of this is a hydrolysis reaction, which means you're going to break down bonds. It's the reverse. So instead of forming water, you're going to take water and throw it in the mix. Okay? So you take water, hydrogen forms to one monomer here, and then the hydroxyl group to the other, and it splits it apart. So here's a real life example with sucrose. Sucrose is a sugar, but it's actually has two glucose molecules bonded together. And what we're going to do is we're going to add water to it, maybe an enzyme sucrase to split water, add the hydroxyl group to one end and the hydrogen group to the other. So breaking the covalent bond with water. Hydrolysis. So if you're struggling to remember, okay, so condensation means you're going to form water, but hydrolysis, lysis means to split or to break down. Okay, and then hydro is water, so water splits or breaks the bond. So, picture just showing between the two reactions. Dehydration, I make water and form the bond. Hydrolysis, I add water and break the bond. Okay, moving on to carbs here. Energy storage and structural molecules. A carbohydrate usually has a 1 to 2 to 1 molar ratio between carbon, hydrogen, and oxygen. So that's one way you can identify a carb is this 1 to 2 to 1 ratio. So notice here sugar, C6H12O6, it's a 1 to 2 to 1 ratio. So this is another way to simplify it is carbon, water, and then the end would be the same. So like, just to try it out. So if I was talking about glucose, I have 6 carbons here, and in this case, 6 water molecules. And if I FOIL factor, FOIL sounds okay. FOIL distributes, okay. But anyways, this 6 goes to here and to here. So then I have C6H12O6.
But N means 6.
Yeah, because N is like in math, you can plug in any number usually, isn't it?
Well,
Right?
yeah, it's just a variable, so you're plugging in 6.
Okay, all right. Just want to make sure we're on the same page here. It's not. If it was a capital N, it would be completely different. It would be okay. But carbohydrates, one of the characteristics that carbohydrates have is they have many CH bonds, and we talked a little bit about it yesterday. How maybe it was 2 days ago. I'm not exactly sure. But see, if you have a lot of CH bonds, you can break them or form them and release and absorb a lot of energy. So simple carbohydrates, a monosaccharide, which is a glucose molecule, ring-like shape here. I can take a glucose and add another glucose to it and form sucrose. We call that a disaccharide, prefix di for 2 saccharides bonded together. So another word for sugar is saccharide, so keep that in mind when you see saccharides. And then if you get a long chain polymer here, we usually call it a polysaccharide. So many sugar molecules bonded together. So monosaccharides are your simplest sugars. They can exist as few as 3 carbon sugars to 6. 6 obviously plays a central role in energy with glucose. 6 carbon sugars can be straight-like chains, but in an aqueous state, they will form rings. And so there's a picture in your book, figure 3.6, that kind of shows the two there. The empirical formula, you can write it out as C6H12O6, or if you want to do the simplified version of it, with the CnH2ON, there it is right there. But glucose is not the only sugar with this formula. So I know in biology 10, we mentioned sugar and glucose kind of as the same thing, but really there's a lot of different sugars out there, and glucose it's just not just glucose here. So we'll take a look at some of these structural isomers, okay, like glucose, same chemical formula here, C6H12O6. Glucose actually has 2 forms, alpha, so whenever you see kind of like this Jesus fish symbol, okay, alpha, yes, alpha and beta. So the 2 forms of glucose are actually fructose and galactose, and the only difference between these 3 right here with fructose is that the carbonyl carbon, it's in a different place, and in galactose, the hydroxyl and the H groups are in a different place. So I do have the structural isomers pictured at the bottom here. Here's what glucose looks like. 1, 2, 3, 4, 5, 6 carbons, and then this is an oxygen, and you can count them all up. It will be C6H12O6. Now fructose, 1, 2, 3, 4, 5, 6. Okay, but then notice that the shape of the ring.
There's an outlier. There's a little extra line.
Yeah, there's a little extra. Yeah, so it goes one, two, three, four are involved in the ring, but then this one has a carbon branched down and a carbon branched up. But I still have 6 carbons, and that oxygen to kind of connect the ring. But same chemical formula. So notice the shape here. We have that nice little hexagon, and this one's a pentagon. All right, now galactose. Okay, we have the hexagon shape. Let's count our carbons. 1, 2, 3, 4, 5, 6. And does anyone see where that difference is?
It's not a structural thingy, but it's a.
It's got the H.
It's a. Hold on. What? A styro.
It's upside down.
It's a
upside down.
because it's a mirror image.
Is it?
I don't know. That's kind of what I thought. But
No, it's not. Okay,
Shut you
But-
down.
but notice
almost.
this one group is just flipped. Just- All right, functional differences between the isomers as a result. They do have some different properties. Galactose is usually a milk sugar. Fructose is a sweetener. There are different enzymes that act on these sugars. Okay, so another diagram. This one's more color-coded, if you will. So here's glucose, and they emphasize the double bond where it's at, and then that flip between the hydrogen and the OH group. So in fructose, the hydrogen is in the same place, but this double bond is somewhere else. And then in galactose, the double bond's in the same place, then these guys are flipped. Okay, disaccharides. What's disaccharides again?
Sugar.
Right? What's
2 of them.
2 of them bonded together. The main purpose of disaccharides in plants is to transport sugars within the plant. So plants can't move monosaccharides. The only way they can move sugars is actually in the disaccharide form. And then in disaccharides in animals, it's to provide nutrition. So humans move glucose as a simple monosaccharide. Plants have to convert it to a disaccharide so that way it doesn't get broken down right away. Because plants tend to store their sugar and kind of save it for a rainy day. Made by linking 2 monosaccharides together to form that disaccharide. In your book, figure 3.8 does a nice job showing that. If you were to take glucose and fructose and bond it together, because this is a monosaccharide, this is a monosaccharide, I form it a disaccharide, but in this case it's sucrose, which is table sugar. So I don't know if your minds are kind of, blown right now, because you always think glucose is just sugar, like, oh, I'm eating sugar, or I put sugar on my cereal or whatever. Well, it's a different form of sugar. It's actually, you're eating a disaccharide. Okay? So sucrose is actually table sugar. Glucose, monosaccharide, plus galactose, another monosaccharide, bond together. You get lactose, which is the milk sugar. So if you are lactose intolerant, that means you lack the enzyme, or your enzymes in your digestive system are faulty and don't recognize lactose. And
Are
then
those the only 3 mono, whatever? How do you know if they're like the mono?
oh no, there's
saccharides or-
more.
How do you know?
But these are the big ones. The big
Wait,
ones.
what's a one sugar? Is that what monosaccharide means?
Yeah, just one sugar.
Oh.
Okay, if that wasn't enough, we even have maltose, which is 2 glucose bonded together. We already talked about sucrose. What's sucrose again?
Sugar.
Table sugar, and then lactose. Where do you find lactose? Milk.
So what's the maltose?
Maltose.
Malto,
Well, you're not too
It
far
comes
actually.
in the- when they
It's
make
in
the malt.
grains usually.
When they come to malt drinks, that's why they call them malt. Is that extra ingredient?
Okay, so polysaccharides provide energy storage and structural components. So many saccharides bonded together, and we make it through the dehydration synthesis reaction. Some examples of polysaccharides: starch, usually made of alpha glucose molecules in a very long chain. And then we are also going to talk about cellulose, how it's made of beta glucose chain. So we're going to talk about the difference between alpha and beta glucose here. Okay, so this diagram shows glucose molecules, but they're arranged in a different way. So I'm going to start with cellulose, okay? These are all glucose molecules, but what do you notice about the arrangement of the glucose molecules? Just look at the top row here.
Oh, they're flip flop.
They're flip-flopped. Okay, so let's just say that my pinky is the CH2O group, all right? And now it's down here. So they're flipped. Up, down, and because of that, that's why we can't digest cellulose. We have enzymes that will recognize this, but then when they get to this part, they're like, what the heck? I thought you were this. So that's why you can't digest cellulose. So when you eat celery, when you eat corn, it doesn't really get digested.
Wait, so are those styrosomers with the flip?
Okay, starch. Take a look at this. What do you notice about starch?
They're not what?
They're not flipped, okay? They're just boom, 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 okay? All right, glycogen. H-O.
Oh wait, that's the same on the other one too. Never mind.
That's just the leaf.
Is there any difference?
They're all the same.
They're all the same. So I think a lot of it has to do with where you find it in the organism. So. But glycogen in animals, this is how we get our energy. It's stored in that form. Starch is plants. Okay. All right, I'm going to throw in chitin.
That one's flipped.
It's flipped and
It's like
Backwards.
It goes back
No.
backwards.
It's long.
Doesn't it?
it's flipped and there's a special group added to it. Does anyone see the ends? All right. Yeah, it's a special group of atoms bonded together. Chitin, very durable, strong structure. You find it in insects. You find it in the walls of fungi. Fungi don't have cellulose; they have chitin. So chitin's a kind of another,
Of course, fuck.
molecule that doesn't really get broken down very easily. So. All right, let's talk about starches and glycogen. So organisms store metabolic energy in monosaccharides, and then we convert them to disaccharides. If you continue to link maltose together, eventually you're going to get starch, which is an insoluble polysaccharide. What does insoluble mean?
It can't be solid. It can't be breaking down.
Say that again.
Not able to be.
Okay, yeah. So insoluble, you can't really mix it with water. It's got some non-polar characteristics to it. So one example with starch in plants is potato starch. The simplest structure of starch is amylose. Hundreds of alpha glucose molecules linked into long unbranched chains. With potato starch, it's 20% amylose, and it's got the alpha 1,4 linkages. Now these numbers here denote the carbon number in the molecule, showing you where the bonds are at. There is a more complicated variant of amylose called amylopectin, where it's branched off into many places. So with potato starch, I told you 20% is amylose, but 80% is amylopectin, more of this complicated starch, and it's linked together with alpha 1,6 linkages. Now I probably just said a bunch of mumbo jumbo, but I do have a diagram on the next slide that's showing this alpha 1,6 linkage, showing the complicated branches and just unbranched chains.
Is there some polysaccharide? Is that when there's a lot of different sugars?
It's a bunch of monosaccharides bonded together.
So it goes like mono, di, poly.
Yes,
But it's still sugar, right?
still sugar. Yep. Okay, so here's amylose. I forgot to mention that alpha means that it's like upright, and beta is like when you start to get it flipped upside down, if you will. Sorry about that. So anyways, alpha 1,6, if I were to count my carbons here, 1, and then notice I have 2, 3, 4, 5, 6. So here's my 1, and here's my 6. So those are the ones I'm going to emphasize. Oh no, I did this backwards. Hold on. Oh, they have it counted different. Okay, so actually they count it backwards. My bad. 1, 2, 3, 4, 5, 6. My apologies. So then over here would be 1, 2, 3, 4, 5, 6. Okay, so here's the 1, here's the 4. Both glucose molecules are upright. The bond is between the 1 carbon on this side and the 4 carbon on the other monosaccharide. So there we go. And you could do that for all of that. Now, for the amylopectin molecule, where it's branched out, so here I have a straight chain, and all of a sudden, boom, now I'm just going to just make it even more messed up. Branch it off. I'm going to use an alpha 1,6, so in that case, it would be a 1, and then it would be attached, if I could shift this down, 6. That's where the bond would be. Questions about, do you have any questions?
I feel like I didn't really understand that.
Yeah,
Be able to have a
I feel
question.
like I don't really even know what to ask. So.
Yeah. Okay,
Just this slide was kind of
all right, so do we understand that alpha is upright?
Yeah.
Okay, and do we understand that the 1 and 4 are counting carbons on the
Yeah.
monosaccharides?
How is there a 5 and a 6 on the same line?
5 and the 6 on the same line.
Like
Okay.
on the same, isn't that like the same carbon thing?
Okay, I don't know what, do you want to come up and point
Well,
it
just
out?
like where it says like when you go 1, 2, 3, 4, and then there's the 5 and there's the 6 above it. Isn't that on like the same line?
What do you mean by the same
what's a
line?
different point on the line.
Oh.
Remember points where lines meet is a carbon atom.
Oh, okay.
So
I
it's
didn't
probably
know
like
that.
on
Do you think or
Like a
do
Q-
you have it?
like a Q-tip.
It's like
Coffee cup.
A Q-tip.
Like,
There's 2
each one of
sides
this
to
is
Q-tip.
this is a carbon, this is a carbon. So it's like, okay,
I got it.
you're good?
Yeah.
Okay. All right. Now, glycosidic bonds are just saying this is the type of bond that forms between 2 sugar monosaccharides. Okay. So that's a prefix for sugar. Yeah, so it's just a sugar bond. Is that the? Oof. All right. With me so far. So alpha, because it's upright. Amylose is just a straight chain starch molecule in plants. That's it. However, they also have the ability to make it more complex by adding side chains. In this case, right here. So it's still a straight chain amylose molecule, but how they make it branch is instead of adding it as 1,4, because if I did a 1,4, then it would be straight chain, it would just be amylose. But they have to make it branch, so it has to attach to a different carbon, in this case, alpha 1,6. So let's just say I wanted to make, I think I could draw one out here. Let's say I wanted to add another amylose straight chain, but make it branched, so I get the amylopectin. So then here, I'm going to attach it to the 6 carbon, and this is the 6 carbon that I labeled over here. So it would be CH, no, it would be CH, because remember when we form bonds, we're going to release water. So there goes that hydrogen, there goes that oxygen, and there goes that hydrogen. So it would just be CH, and then I'd go down, and I'd make my little hexagon. And then OH, there's a branch there. Okay, and now I would just add it, and now I would start to go down.
So all you did right there was drew the exact same thing, but connected at 6 instead of 4.
Right. You good here? Okay, I guarantee you guys there is a multiple choice question on the exam talking about amylose and amylopectin and what kind of bonds are associated with them. So amylose, alpha 1,6.
Glycosidic bonds.
Okay, amylopectin, alpha.
Pectin is 1,6.
Yeah, you said
Shoot,
that.
I said that backwards. Ugh. Okay, but yes, understand the bonds between the two. Gotta know them.
So they just
Oh,
connect.
like why does this happen? Like why would it
Okay,
bond with a 6 instead of a 4?
well, because it's branched, so then enzymes can't recognize it and break it down. So then that's why potatoes, you know, you can smoke potatoes forever before they start to get all. So long-term storage.
Does it only release a water molecule on the H6 or on the 1,6 for the amylopectin, or does it do that for the amylose too?
It does it anytime you form a bond.
Oh.
Okay. Animals. So we don't have starch. We don't make starch. We make glycogen. And glycogen is comparable to starch in animals, comparable to the amylopectin, so the one that's branched, okay? So glycogen is insoluble, so it can't dissolve in water, has some kind of nonpolar properties, contained in branched amylose chains. Chain length is greater and more branches than the plant starch, so it's even more complicated, even more of a mess. So this is what starch looks like. Here's glycogen. Oh, we start, oh, I want to branch. Oh, I want to branch. So. Okay, looking at the bonds of glycogen. Okay, again, alpha 1, 4. What's this one?
6.
Alpha 1, 6. And I zoomed in here. It's the alpha 1, 6. Straight chain, alpha 1, 4. Branched, alpha 1, 6.
So is there 3 1/6s right there?
3, 1.
The one they have the circle on, one that's
That's
in
2, isn't it?
one, yeah, one there.
Alpha 1, 6, alpha 1, 6.
Yeah, and then there's one.
I mean, alpha 1, 4, alpha 1, 4, alpha 1, 6.
and then there's one over there to the left.
Oh yeah,
And then there's one up
the
there, I
top.
guess, too.
On the
Oh, yes,
bottom.
that's branched. Yep.
And then
That's
also
another
the one
branch.
right there, so it'd be 3.
Yeah, so 1, 2, 3. Yes, there's 3 alpha 1, 6.
There's a lot of them.
Okay, cellulose. So cellulose is glucose molecules of the same form bonded together to form that beta 1, 6. Now beta means they flip-flop. Upright, upside down. Upright, upside down. But it's still beta 1, 6. So again, you will see an exam question with cellulose. And what bonds are associated with cellulose? So beta 1, 6. Long unbranched beta-linked chains are tough fibers, very similar to amylose, except that they're upside down. So they're not alpha. Okay? They're beta. And as a result, there's enzymes out there that cannot break these bonds. That's why cellulose just doesn't get broken down very easily. There are a few animals out there that can break it down, but they have to have bacteria inside their stomachs. Protists, there are some protists out there that can also break down cellulose. Matt.
Wait, so is it beta 1/6 or beta 1/4?
Did I say beta 1, 6 again?
Is it
Yeah. You said it like 4 times in a row. I was like,
I'm so sorry.
Wait, so that's a beta, right?
This is beta 1, 4. I'm just, I'm so
But
sorry.
so in like amylose, that's alpha.
Yes, amylose is alpha 1, 4.
yeah, and you were like reiterating it each
Ah.
time, so I was like, oh.
Okay, so looking at a leaf, zoom in on the plant cells, you see the cell wall. Zoom in on the cell wall, you see these fibers kind of intercrossing. Pull out one of those fibers. Fibers are made up of even more individual strands. And then eventually you're going to start to see that string of glucose molecules arranged in beta formation, linked together, beta 1, 4. There we go.
So those would be like the sisomers, kind of, because they're going back and forth when it's alpha beta.
Okay, so then starch, straight chain, alpha 1, 4, cellulose, straight chain, beta 1, 4, glycogen, okay, the straight chain part is alpha 1, 4, but then to form the branch,
6.
alpha 1, 6.
What things like do alpha 1, 6? Like can anything just do that, or is it like?
It's kind of a characteristic with carbs. Okay, last thing I want to talk about here is chitin, which is similar to cellulose. Okay, it's a modified form because you get the beta kind of going on here with the upright, upside down. However, with chitin, there's this N-acetyl group, and so this is what N-acetyl looks like. On the previous picture, it just had N, and you guys said nitrogen. That's okay, but it's an N-acetyl group. It replaces a hydroxyl group. So it's cross-linked with proteins. It's very tough, resistant surface, kind of water repellents. That's why you find it on the exoskeleton of insects as well as your crustaceans. So then, this is the slide I showed you before, and that's it for for carbohydrates, really. So know the linkages between these molecules, and understand monosaccharides form disaccharides, the polysaccharides.
Wait, so what's chitin again? So it just
Chitin?
flips the N and then it changes, or then on the end.
All right, so chitin is beta,
Oh, okay.
1,4, but instead of a hydroxyl group like up here, it's that N-acetyl group.
Oh,
So it's a modified form of cellulose. It's just that N is.
So those would be polymers, right?
These are all polymers.
Okay. So the glycogen polymer right there is 1,4.
Yes.
Okay. But on this yeah.
Oh, it's branched. Yeah, but they didn't want to mess up.
Okay, but on this one, so glycogen, do you actually call it 1,6 even though there's a bunch of 1,4s?
No,
because
it's-
has a couple.
So the straight chains that you see are 1,4s, but then when you see it branched, which is glycogen, it's
Yeah.
it's got
So
branches.
the actual name in general is glycogen.
Combination of both.
But yeah, combination.
Okay, so I'm going to stop it right here. So tomorrow we'll talk about nucleic acids and basically energy storage molecule. We'll also talk about a new hypothesis called the RNA world hypothesis. So maybe RNA was the genetic molecule before DNA came onto the scene.